Hi, hello, morning to all. Today topic is complementary DNA or cDNA. We are all know that DNA, RNA, plasmids. But what is complementary DNA? Means the specific type of DNA to be produced from mRNA that produced cDNA, which is used for cloning. That is called cDNA or complementary, because complementary means produced DNA complementary of isolated specific mRNA. That's indicated complementary DNA. Moreover, prokaryotic and eukaryotic gene cloning both are different type of gene structure. So on coming slides, we can learn. Let's go. cDNA always produced from isolated or purified mRNA. Why we choose we say mRNA for producing complementary DNA means it's behind a beautiful principle is that. What is that? We something know that prokaryotic gene structure or prokaryotic gene expression based on operon concept. Operon means a cluster of genes that encodes different type of proteins, that proteins to play the role in prokaryotic system. So the cluster of genes are linked together. That cluster of genes are controlled by a single promoter or operator or regulatory sequences in prokaryotes. So the entire concept is operon concept. But in eukaryotic system, one gene, one enzyme hypothesis are followed. Of which individual protein were synthesized and produced from single gene. So here a single gene was controlled by single promoter or single operator or other regulatory sequences. But prokaryotic system, multiple cluster of genes are controlled by single promoter or operator or other regulatory sequences. In prokaryotes, the cluster of genes are produced polycystronic mRNA. That means a single mRNA get translated to produce more than one number of proteins. That is called polycystronic mRNA. But eukaryotic system, single gene produced single mRNA. A single mRNA encodes single protein. So one gene, one enzyme, hypothesis. But here in genetic engineering, we try to clone any kind of genes. So we must have the complementary DNA for cloning. The complementary DNA is called coding DNA in the prokaryotic family. So mRNA, by using reverse transcription, produced single stranded DNA, then followed by single stranded DNA with the help of polymerase polymerase enzyme produced complementary DNA or cDNA. It's become double strand. So single stranded mRNA converted to single stranded DNA, then it's converted into double stranded DNA. So in that double stranded DNA used for gene of interest of cloning or prokaryotes. Prokaryotic gene structure. So I already told that is a operon transcript. Operon means a cluster of genes. For example, X, Y, Z. A cluster of genes are linked together. These genes are controlled by operator and promoter and other regulatory sequences. The famous lac operon, trip operon, ara operon are working like this. So these cluster of genes are produced single mRNA. The single mRNA produced more number of proteins. Next one is eukaryotic gene structure. It's very different from the operon prokaryotic gene structure. Here, a single gene from start point to end termination point is a single gene. A single gene encodes single mRNA. A single mRNA is called monocystonic mRNA. That mRNA produced a single. But why we need the complementary DNA for cloning? Here the secret is the eukaryotic gene containing introns or exons. Introns are non-coding sequences, exons are coding sequences. So the introns are coding to encode the proteins, but the introns are non-coding sequences, not encoded in the transcription or protein synthesis. One more is poly A tail sequences are there. It's a unique conserved sequences present in all eukaryotic mRNA system, which is used to export the mRNA from nucleolus to cytoplasm in eukaryotic system, and also provide the stability of the particular mRNA until the translation is complete. One more site is there, it's called termination site, which is containing stop codon UAA, UAG, UGA. So the specific conserved codon present in the sequence. So it's play to stop the transcription process. One more sequence is there, it is called start codon or AUG codon. So the AUG codon used to assemble the ribosomes during the translation, and it provides some other functions also. Come to the point. Why we need complementary DNA for cloning? That means we are using variety of number of plasmids depending upon the gene, depending upon the DNA size. In primary stage, we carry approximately 100 kb size of gene. So, the gene of interest, maximum size of gene of interest is approximately 100 kb size. But in the particular gene, which gene we want to clone, that gene contains more number of introns. So we must remove the introns. Then only to reduce the size of the gene or gene of interest and make it to cDNA per clone. How means? First of all, we need collect the specific mRNA from which gene we want to clone. Suppose we collect the total RNAs from the given sample or given tissue or given plant tissue, whatever it is. First we isolate total RNAs. Then we separate the tRNA, rRNA, and mRNA using the chromatography technique. Finally, to remove the tRNA and rRNA, we have group of mRNAs. That means the mRNA, group of mRNAs indicates how much of number of genes are expressed at the particular time of we collect the mRNAs. Okay, but we need single gene. So we once again separate the single gene mRNA from the total mRNA. So that specific mRNA used for synthesis of complementary DNA and are otherwise called substrate for synthesis of cDNA. That mRNA contains no intransigence, only exons. Because eukaryotic system to involve or manipulate the mRNA, the excess non-coding sequence or introns are removed by splicing reaction. So the splicing reaction is important. Reaction takes place in the eukaryotic system to remove all intron sequence from the synthesized mRNA. So we collect that mRNA for synthesis of cDNA. And how we will generating the cDNA library. cDNA library means the single cDNA contains more than number of one genes. So the group of genes which indicates cDNA library. Suppose a single cDNA containing single genes is called complementary DNA. That's all. But the single DNA contains more number of genes. So this is an additional process, combination of one or more number of cDNA to produce complementary library. Similarly, genomic libraries differ from the cDNA library. We will learn later. The entire genome was isolated, randomly cut by the transcription enzyme, immediately recombinant to the vector, and the vector transformed into the host cell and finally expressed. So this is a crude method for construction of genomic library, but cDNA library is advanced method from genomic DNA library. Okay. Here now, how to construct the cDNA library? I already told that all eukaryotes have an mRNA as a monosystemic mRNA. Okay. Each specialized cells have specific mRNA that encodes information for a specific protein. So one gene, one enzyme hypothesis. Okay. This information can be transformed back into DNA in other words, a DNA copy of mRNA. It's called complementary process using reverse transcriptase, a specific enzyme which plays convert the mRNA into DNA, single stranded DNA. This DNA is called complementary DNA. This complementary DNA stored in plasmids or phage nature. The cDNA contains only the expressed genetic information. There is no introns, only exons are exist, which allows to study the amino acid sequence directly from the DNA. So the all complementary DNA sequence to encode the corresponding amino acids by the genetic code probable hypothesis process. Okay. First step in creating a cDNA library. First to isolate the total mRNA from the cell. Here the process detailed described. Here oligonucleotide matrix. This is a type of chromatography. It's a stationary piece. The polythymine, oligonucleotide thymine, oligo primers are tagged with the stationary piece. The entire process is called affinity basis to play the role. And finally, we add the cell-free system to the chromatographic column. It contains tRNA, mRNA, RNA. Yes. So removal of the tRNA and rRNA, because it's no have the specific poly-A sequences. So the poly-A sequences are correspondingly bind to the polythymine oligonucleotide primer. So apart from that, the unwanted rRNA and tRNAs are eluted out. Once again, we change the ionic strength of the chromatographic column. We eluted the total mRNA from the cell-free system. It's a substrate for cDNA synthesis. So all mRNA have poly-A. By using the column chromatography or other chromatographic techniques that contain short polythymine sequences as a stationary piece, to use to separate the total mRNA of the given cell-free system except tRNA and rRNA. After separation of mRNA, the mRNA treated with the reverse transcriptase enzyme. So the enzyme to add the complementary to add the nucleotides finally produced single stranded complementary DNA. Then produced RNA DNA hybrid. RNA DNA hybrid is produced. So on the specific RNA, mRNA was digested using alkali. The mRNA is removed. Finally, single stranded cDNA is exists. So here it will describe mRNA having poly-A and hybridized with oligonucleotide primer, which is present in the chromatographic column. So specific polythymine primer complementary binds to the poly-A. So remaining unwanted tRNA, rRNAs are removed. Finally, RNA, DNA hybrids are produced. Then the removal of mRNA using alkali, add some alkali solvent, digested mRNA, because mRNA lifetime is very low. Example, eukaryotic mRNA lifetime is exactly 5 minutes. So finally produced single stranded cDNA. The single stranded DNA again treated with the oligonucleotide primers with help of DNA polymerase enzyme. The single stranded converted to double stranded. The double stranded cDNA is suitable or coding sequence for that produce the recombinant DNA. Then this is gene of interest. Gene of interest. The interest then join the corresponding vector. In genetic engineering, we kept the important point is when you are start involved in the gene cloning procedure wherever. Suppose you choose gene of interest, but gene of interest, the vector, both are treated with the same restriction enzyme, especially type 2 restriction enzyme, especially sticky end producing restriction enzyme. So the gene of interest and vector both are treated with restriction enzymes, same restriction enzyme, and produced same sticky ends. The same sticky ends also complementary to each other and easily recombinant molecules are created. So like this, ligate with the corresponding cDNA with the linker. The linker is additional sequence learned in detail. Suppose the linker added in the case of the cDNA when it's blended. So finally converted to sticky end with the help of linker or without help of linker. So that is the ready gene of interest to ligate to vector. Then packaging of cDNA. It's a very interesting procedure. We produced cDNA library for gene cloning. So the DNA have low stability, so it must be packed with the vector or host page. Usually, the cDNA, synthesized cDNA, immediately packed in the lambda phase or M13 filamentous phase because both are effective vector to inject the DNA into the host. So the DNA, suppose produced cDNA have blended, we add the linker, then the linker treated with the enzyme produce the sticky ends. Now the sticky ends are complementary to each other, the vector to produce recombinant molecule. Why we need the packaging of cDNA means cDNA. Suppose if we start a cloning experiment, we just insert and clone the one gene. It is very complicated process. But at the same time, we inserted or cloned more than one gene at the same time. Both are same procedure. Why we use single gene clone? So we take a multiple genes to done the same procedure, and it may do number of genes product if you want for the application purpose. So the producing single standard or single gene complementary DNA, that complementary DNA once again join together to produce cDNA library. That's we need packaging with the help of lambda phase or M13. M13 filamentous phase or lambda filament phase used to construct the cDNA library because the genome lambda phase M13 filamentous phase genome is very useful packaging the cDNA because it's containing specific sites, example cos site. So similar sites are there. Suppose we no need to complementary library, we need a single cDNA gene means, and also construct the probe sometime for screening of clones or synthesis of oligonucleotide primer or a labeled primer. So at the time we obey the radio labeling technique, which is used to screening the corresponding gene product present in the cloned host or cloned recombinant vector and priorities. It's an essential DNA probe needed for screening. For that purpose, the synthesized cDNA involved into labeling process. First, we select the double stranded cDNA and treated with denaturation or nuclease dilution and produce a single stranded cDNA. And the cDNA once again they treated with the alkaline phosphatase enzyme. So in alkaline phosphatase, remove the 5' and cleave the 5' or 3' end of the DNA. Same time, radio labeled nucleotide triphosphates added to the specific DNA sequence. Mixture and also help of DNA polymerase. The labeled DRC nucleotide triphosphate added to the single stranded cDNA. So finally converted into labeled cDNA, or sometimes produced labeled cDNA library. Thank you for watching. Thank you.